Again, my name is Sato. I'm the CSO of Twist Bioscience. Today I'm going to give you a really brief overview of all the services that we offer. And then since I have an hour, which is really unusual, I'm going to spend the time going through 6 case studies, so some really nice stories, kind of end-to-end projects with utilizing those services, and I'm going to, again, show a lot of data associated with those. So rather than kind of keep it high level and talk about all the great services we have, I'm going to kind of keep that short and spend a lot more time talking about stories with data. So you may not be familiar with Twist Biopharma Solutions. We're actually a division of Twist Bioscience, which, as you may know, is a very large established DNA synthesis company. Twist Biopharma Solutions, we rebranded ourselves earlier this year because we were basically merging 2 groups together. One was the kind of legacy Twist Biopharma team that I helped found a little over 5 years ago that focused on using DNA synthesis and libraries to make in vitro display libraries in phage display. And then about 2 years ago, we acquired a fantastic team in Boston called Adveris, which had a lot of expertise in vivo antibody discoveries. So the combined organization now has the capabilities to do both in vivo as well as in vitro antibody discovery. And at the same time, we also brought online a lot of computational and in silico tools to really round us out and really extract more value from our data. So that's why we wanted to rebrand ourselves as a new unified team. I'll talk today again at a high level about all the services that we provide in those 3 key aspects I just went through. And you'll really see the power of having each of these 3 kind of capabilities within the team to enable us to make sure that you're successful in the end in discovering and optimizing antibodies. So I'm often asked, Aaron, what differentiates Twist Biopharma Solutions from your competitors? Great question. The first one I love to cite really goes to those kind of 3 technology services that we provide, in the sense that we can generate antibody diversity from a wide range of sources. As you'll see, we can discover antibodies from in vivo sources, in vitro sources, and then we can use our AI and machine learning to help us find even more hits from our data. So that really gives us a huge advantage over a lot of our competitors. The second is really the depth of how far we can go into our screening. Since we actually sit inside of a large DNA synthesis company, we're not limited on the make-test part of our discovery process. So if we need to make hundreds, maybe thousands of antibodies derived from the projects that we do for you, we can easily do that because we're not limited on synthesis or the production of antibodies or the downstream testing. And then finally, we're really great at doing not only discovery, but also optimization and antibody engineering of leads. I'll talk today about some of the antibody optimization services we have, as well as the work that we can do around humanization, as well as some work that we're right at the cutting edge of showing, which is our ability to use language models for increasing the affinity and stability of antibody leads. So getting back to those kind of 3 key aspects of our team, first one being in vivo immunization. I'll show today the power of the Adveris system that, again, we acquired 2 years ago that sits in our Boston facility. We have our own proprietary mouse, which is still on a wild-type mouse background, but it has several key mutations in it that enable it to have a very open immune system. And so this actually allows us to discover antibodies to highly homologous epitopes that have a high level of human-mouse homology. And so in many cases, oftentimes we can actually discover antibodies that have this human-mouse cross-reactivity, and we don't actually have to go to a knockout model, for example, to get those antibodies. The team also has a huge amount of expertise in running the Beacon. We can do single B-cell cloning, not only with our proprietary mouse, but we also can do immunizations in other species, such as alpacas. And we're one of the few companies that actually can do single B-cell screening coming out of the alpaca. And so that's, again, a nice differentiation compared to some of our competitors. And then finally, we have a lot of expertise in different immunization strategies, particularly in our mouse. I won't show it today, but again, we have the ability to do both DNA, cell, protein, peptide-based immunization, as well as a lot of proprietary adjuvants that we can bring to the table. On the in vitro side, as I'll show, we have over 20 antibody libraries, all in phage display. We have examples of Fab, single chain, VHH single domains, common light chain. So really the nice thing about, again, these different formats is you can be really modular in terms of how you do your panning and your screening, which enables you to come up potentially with different formats against different targets that you could potentially use for making, say, bispecifics or multispecifics. And as I mentioned, this team also, again, sits inside of the larger DNA synthesis organization of Twist, so that gives us a lot of power for that make test cycle that I talked about before. And then finally, I'm really excited about a lot of the AI and machine learning we're doing in the team. We have a small but nimble team inside our South San Francisco facility, and they're really helping us to maximize the value of our data, get more potential hits from our data, as well as to optimize sequences and actually come up with new mutations within some of the sequences that we generate. So let's start with, at a high level, our in vivo offering. As I mentioned, we have our proprietary hypermeme mouse. It's actually put on two different backgrounds, BALB/C and BLAC6. As I mentioned, it has several key mutations in it that enable it to have a very open, low level of tolerance, which enables us to find antibodies to highly homologous epitopes between mouse and human. As I mentioned, we're also actively doing a lot of work in alpacas to discover heavy chain only antibodies. I'll show some of that in one of the case studies that I'll talk about today. And we're starting to explore other species like rabbits and maybe even potentially chickens. Again, all utilizing single B cell cloning, except for the mouse work where we can actually do both. As I mentioned, we have a lot of expertise in different immunization strategies. I often say that the most difficult part of any in vivo project is actually getting to that titer in the animal, and the team has a lot of expertise in using different forms of the antigen as well as different immunization strategies and adjuvants to enable us to get to titer. And then finally, because we have all this great expertise, a lot of our customers have come to us with some really tough, challenging targets like GPCRs and ion channels, and the team has a lot of experience tackling those targets and actually being very successful against them. On the in vitro side, as I mentioned, we're doing a lot of work to generate more and more libraries every year. As I mentioned, we have about 20 libraries in phage display. We utilize the Twist oligosynthesis platform to enable us to make these libraries. And then we can do panning and screening for our customers to pull out specific sequences against these targets. Again, unlike the in vivo approach where we have to basically generate a big titer against the target and then screen for the potential hit, here using phage display, we can actually pan out and find the desired specificity that we require utilizing these libraries. And oftentimes we'll pool them based on scaffold type to enable us to find very, very specific binders to our customers' targets. So I love to use phage display when our customers come to us with a very specific site they want to hit or want to have a very defined specificity requirement because we can really take advantage of the power of display and selection to find it. And then finally, again, this just goes through our process on the top level of the slide. We really have a lot of flexibility in terms of how we do our panning and screening. And then as we get sequences through either phage-based ELISAs or through the AI machine learning approaches that I'll talk about in a second, we're not limited on that make test. We can immediately synthesize all the leads as either IgGs or VHHFCs and then go on and do the downstream screening. So then finally, on the AI in silico side, we're doing a lot of work to really expand, again, and get more value from our sequences that we generate from both our in vivo and in vitro approaches. We're actually using a lot of our AI to help with library design. We can actually use our large antibody databases and models of those sequences to actually feed them human germline frameworks, and then actually ask those databases to fill in the CDR sequences within those frameworks. And so we can actually use that as a way to generate alga pools and diversity within each of those germline frameworks. And that enables us to then use our AI to help us with library design. Another big area that we use AI for is that when we do our panning and screening that I talked about in the previous slide, we can do NGS analysis of those multiple rounds of panning, and then we can use multiple, both unsupervised and supervised machine learning to enable us to pick out hits from that data. And so rather than having to rely solely on ELISA-based screening, we can actually use NGS and these tools to help us pull out even more hits from our data. Finally, thirdly, on the humanization side, we're doing a lot of work as well to use AI and machine learning to help us with creating rapid pipelines for humanization. I'm also really excited about how we're using language models, both zero-shot and few-shot models, to help us come up with variants of specific sequences. And this is really allowing us to rapidly move forward in optimization processes and not be so reliant on library-based approaches. And then finally, we've actually accumulated a lot of antibody developability data over the years and specific assay data. We can actually use that assay data to create developability predictors, where we can actually feed a sequence into those predictors, and it'll actually give us a potential result. And we've seen really nice correlation between the experimental data and the predicted data for these assays. And we initially started with HIC, but we plan to go into other assays as well. And then on the optimization side, as I mentioned, we have a whole pipeline now for doing rapid antibody humanization, and as fast as 5 to 6 weeks, we can take your mouse or even say a VHH from an alpaca or a llama and rapidly humanize it. And so we come up with multiple graphs in different frameworks, and then we do a small matrix of heavy and light chains. We make all the resulting antibodies, and then we do downstream affinity analysis and compare that back to the chimeric antibody. And I'll show a little bit of data on the next slide showing you that service. And then on the antibody optimization side, since we do have this amazing library capability to shuffle natural human diversity in the context of a germline framework, we can do the same thing as we optimize your candidate. We can take your initial hit, which let's say it's a humanized antibody. We can feed that into our large antibody databases, and it actually can give us CDR sequences that are similar within a defined mutational distance that we can set. So say, for example, we give it a distance of 3. We can actually pull in all of the naturally occurring human CDRs that are in our database that are within the 3 mutations of that parental antibody CDR sequence, and then we can use that as a way to design a library around your specific lead. And then we can go through the same panning and screening process I talked about before that we do for phage display. Just a little bit of case study data around our humanization pipeline. This just shows you 3 examples. We actually took 3 clinical humanized antibodies. We went all the way back to the parental mouse antibody and then rehumanized it, and then compared our best humanized candidate to the candidate that was currently in the clinic. And in all 3 cases, we were able to get a humanized candidate that rivaled the affinity of the original mouse antibody. We've continued to do this for our customer projects, and almost every time we've gotten really close to the final candidate. In some cases, again, I don't know exactly why this is, we actually get improved affinity over the parental antibody. So finally, just to wrap up kind of the high-level overview of Twist Biopharma Solutions, we're really excited about a new service that we're offering called our Gold Standard. It's really this idea that we can combine all of these different technologies together in one offering, where a customer could come to us with their target and a very specific design criteria, and then we can then utilize multiple technologies at once, and we feel so confident that we'll be able to address the target and succeed that we actually give the customer a guarantee and a set price for the project. So we find that we haven't had a customer sign up for one of these yet, but I think it's potentially a really nice offering for customers that want to really hit the target with multiple technologies and really enable us to use everything at our disposal to make sure that they're a successful project. Okay, so now I'll jump into those case studies I talked about. Again, I'll go through 6 different data-rich stories, and I'll start off with a VHH case study using our Beacon workflow. This was also published in a paper earlier this year in Antibody Therapeutics. The target I'll talk about today is a VHH campaign we did against PSMA, which, as you may know, is a overexpressed tumor antigen. It's been utilized in a number of different bispecific T-cell engagers as well as ADCs, and we wanted to see if we could come up with VHHs that could be discovered from the alpaca that could potentially be utilized for some of our customers that might want to go after PSMA as part of their ADC or bispecific T-cell engager projects. So the overall scheme for the project is that, again, we'll immunize the alpaca, and with a schedule of from 73 to 216 days, we'll then isolate PBMCs and isolate the plasma from these animals and do titer testing over that time period. We're looking to see both IgG1 initial titer, which again is just indicative of the initial IgG response. But then we also want to eventually see IgG 2-3 conversion, which is indicative of a heavy chain only response. I'll show you some data later when we look at PSMA versus another target, that in the case of PSMA, we're actually able to see nice conversion to 2-3, whereas for another target, we were only able to see initial IgG1 titer. Once we do get titer, we'll then isolate PBMCs. We'll actually push them to memory B cells. We'll load them on the beacon and then do all the downstream screening to pull up and get positives from the beacon. And then we'll sequence confirm, make and test them, and then do all the downstream testing. I'll also show you today about how we can do a rapid humanization pipeline as well, utilizing the humanization technology I talked about before. So let's go into that first phase, which is the immunization and titer. As I mentioned, shown on the left, you can see that the IgG titers are in the solid lines at different time points, and then the IgG 2-3 titers are in the dotted lines at different time points. And as you can see, we start to get nice conversion between IgG1 and IgG 2-3 in the alpaca for PSMA. For the other target, which isn't named on the right side, you can see that we were able to get IgG1 titer, but we weren't able to get conversion to IgG 2-3, and as a result, we didn't discover any heavy chain only antibodies against this target. So the next stage is the beacon-based screening. Again, we did a pretty simple screen on the beacon. We basically assessed whether there was IgG 2-3 secretion for every single B cell in each pen on the chip. And then we also looked for specific binding to, in this case, PSMA. And so we're able to pull out all the B cells that had heavy chain only IgG 2-3 antibodies against PSMA. We then recovered all the B cells that had that desired specificity in terms of 2-3 binding as well as PSMA binding. And then we put them forward for sequencing. Shown here in the table is just the number of hits that we isolated from each of the different time points that we collected and ran on the beacon. And then we also, just showing the top in the logo plot, you can see the level of sequence diversity in the VHH hits, where we actually were able to see a lot of sequence diversity in different lineages within those sequences. We then made and tested the initial parental sequences derived from the alpaca, again, that had specific binding to PSMA. We tested them for affinity to the target. Shown here on the left is just the expressibility and the specific binding of the PSMA clones. And then shown on the right is we did Cartera analysis and looked at the affinities of all these hits. And we're able to see, again, nice high affinity binding, monovalent binding to PSMA. And then we also did an epitope binding analysis where we looked at how many different bins the different VHHs bound to. And we were actually really surprised and pleased to see that we had lots of different bins coming from our VHH campaign against PSMA. And then finally, going back to what I was talking about before, we also generated a rapid humanization pipeline utilizing the technology I talked about before. And utilizing this platform, we were actually able to humanize many of the VHHs that had high affinity binding. We weren't able to do all of them at once in terms of doing just a few germline frameworks, but in general, we were able to see similar level of binding for many of the humanized candidates that we came up with. Okay, so now I'll jump into the next case study, which is rather than taking a beacon-based approach, could we actually discover VHH antibodies from an immune-based library approach? So again, very similar to what I was talking about before with the beacon case, but in this case, we're doing, sorry, it's a little hot. We're doing immunization again in the alpaca, but rather than doing beacon-based workflow, we're actually then going to move forward and make an immune library against this target. And rather than so once we actually can see IgG 2-3 titer, we'll isolate the heavy chain only portion of that repertoire from the PBMCs, and then we'll collect the RNA and then build the resulting library, put it into phage display, and then we'll do panning and screening against the target. Really take advantage of the display platform to enable us to pull out those specific hits. rather than having to screen for it in the beacon. And so again, this just shows you some examples of how we did this for one particular customer target. Again, we went through that same process of library build, and then we had a very detailed panning protocol that we're working on with the customer to get that desired specificity that they require. And then shown on the right is just the number of specific hits that we were able to generate from the panning. And so that really enabled us to really home in and find those specific antibodies in the repertoire that met the customer's requirements. And then once we reformatted them to VHHFCs, we saw a really nice specificity profile from like high affinity binders to weaker binders. The phage display process enabled us to find a broad spectrum of different affinities. And the customer actually wanted to have this because they wanted to have different levels of affinity against their target where they could actually play around with both the epitope as well as affinity as they were building a bispecific. So just to build on that process, we can oftentimes build these immune libraries in tandem with our single B-cell cloning workflow that I talked about before. A lot of our customers love to use immune libraries as maybe as a backup to a beacon campaign. And because we can collect extra blood from the alpaca immunization, this enables us to potentially do kind of a combined approach where we do both single B-cell cloning as well as immune library. And so the fact that we have both aspects of it really enables us to maximize the potential of sequences that we can generate for our customers. And oftentimes when we do both, we actually see different sequences coming out. So really having the ability to do single B-cell cloning complemented with this immune library approach I think is really powerful, and is something that, again, we're set up to do internally for our customer projects. Okay, the next one I'll talk about is a case study, again, around VHH libraries, but now we're going to use the in vitro display libraries I talked about before, which we're, again, utilizing the Twist platform of DNA synthesis. We made several VHH libraries in phage display, and I'll talk about a project today where we use that to discover antibodies to a target involved in immuno-oncology. And that target is DKK1. DKK1 is actually a protein that actually suppresses the immune system. There's actually a lot of validation around this target. There's actually an antibody currently in the clinic called DKM1 from Leap Therapeutics. Thanks. And what we wanted to do is we wanted to create a molecule that had improved efficacy, improved potency over DKN1, and we wanted to use our VHH libraries in a way to see if we could come up with maybe bispecifics that hit different parts of the DKK1 molecule as well. So we utilized a number of our scFv libraries that I talked about before, but also we utilized heavily relied on our VHH libraries. We also collaborated with a couple different AI machine learning companies to get more sequences from our data, particularly for our VHH libraries. And we also did some language model work to come up with variants of some of the sequences that we generated initially from our primary screening. So this slide really speaks to some of those technologies that we brought forward. Again, as I talked about before, we can do NGS analysis of our multiple rounds of panning, and this really enables us to extract more sequences and more hits from our data. What we found is that we got several really high affinity binders that were very functional from our initial primary ELISA screen, but by doing AI machine learning to help us pick out more hits, we were actually able to find a whole collection of other sequences that we hadn't seen initially from our primary screening data. And then, as I said, as I'll show you later, one of our top candidates that we're moving ahead potentially into development actually came from a language model where we actually optimized one of those initial sequences and made it better. In terms of we actually also did epitope binding analysis with many of the VHH binders that we generated, and we compared that back to that clinical antibody that was in phase 2 from Leap Therapeutics called DKN01. What we found is we had two primary bins of binders to DKK1, one that actually was in the same bin as DKN01, but we also found another very large bin against another site on DKK1. And so we were actually able to find an alternate epitope that is not bound by the same, bound by DKN01. So we were really excited to see how this alternate epitope might have different functions relative to the site where DKN01 binds. And so to kind of break apart the molecule and understand more about where exactly these antibodies are binding, there's actually two CRD domains on DKK1s, or cysteine-rich domains. The DKN01 molecule actually binds, it binds, in this particular, it binds to the CRD1 domain, whereas CRD2 domain, whereas our antibodies in the larger bin bind to the CRD1 domain. So we're actually able to break that apart and actually discriminate where those two antibody bins actually were on the molecule. I'll also show you in the next slide, we did some very fine epitope mapping as well to actually show exactly where on CRD1 and CRD2 these antibodies are binding. And that's shown here. This is in a collaboration with Immunoscientific. We're actually able to do a fingerprinting study where we could actually do free radical generation on the surface of DKK1, and then actually add in or take away each of the different antibodies that I just talked about. And this enabled us to, through peptide mapping, to actually determine the specific epitope of each of the different binders to DKK1. And it actually matched up really well with the CRD1 versus CRD2 data that I showed before. We then went forward and did 3 different functional assays against DKK1. DKK1 is actually known to actually block Wnt signaling, but it's not really clear whether that activity is actually involved in that immunosuppressive effect in tumors. And so what we're able to show through some of the molecules that we generated is that binders that bound to the CRD2, that bind to the CRD2 domain, which is the same domain as that DKN01 binds, actually have a huge impact on Wnt signaling. And that if we titrate in those antibodies, we actually see increased Wnt signaling in a reporter assay. Whereas all the molecules that bind to CRD1 have no impact on that reporter assay. But if we look at, say, an assay that looks at immune activation, we can actually see that all the antibodies that bind to CRD1 actually increase immune NK cell activation. And also in a cell-killing assay that, again, is correlated with immune activation, we can also see nice killing against PC3 cells in that assay. So it correlates really nicely that any antibodies that bind to CRD1, we can see nice activation, no impact on Wnt signaling, and nice killing against PC3 cells. So we were actually able to create a pure molecule that only had impact on immune activation and killing and no impact on the Wnt signaling. What's a little odd is that DKN01 actually has some of modest activity in both sets of assays. So we believe that our molecule will have an advantage over DKN01 because it won't have any impact on Wnt signaling. We also, since we had all these VHH binders that bound different places, we also started making some bispecifics that were either biparatopic antibodies that could hit multiple sites, or maybe even antibodies that bound to the same site. We did make one molecule that actually had a VHH that bound CRD1, as well as a VHH that bound CRD2. And so that enabled us to create a molecule that actually was positive in all 3 of those assays that I just talked about. But we also made a molecule that hit the same site, CRD1 site. We took 2 different VHHs and actually combined it into a molecule with 4 VHHs that all bound to CRD1. And as you'll see later, that particular biparatopic antibody, actually it's not really a biparatopic because all the VHHs bind in the same place, but it's a highly avid molecule, and as you'll see in the next slide, that particular antibody has really potent activity in vivo. So when we finally did an antibody, in vivo study, this is again in a PC3 in vivo study in a SCID mouse. SCID mice actually do have NK cells, not T cells, and so you can actually look at immune activation in this model. Again, if you remember back, the cell killing assay that I showed was actually using PC3 cells as well. So this enabled us to do an in vivo study and see how our molecule compared to the DKN01 positive control. And what we found really nicely, we were able to show that we actually had 2 molecules that had improved activity over DKN01. One of our molecules was one of those bispecifics that I talked about before that were both VHHs bound to CRD1. And then we also had another molecule, TB441-37, which is actually a language model hit where we optimized the sequence of one of the binders inside of that other bispecific, and we actually made several key mutations in it to enable it to have higher affinities. So we're able to find a bivalent VHH Fc that had similar activity to the bispecific that had 4 modules that bound to CRD1. So you can really see the power of using our language models to increase the affinity of these antibodies to get higher in potency. Okay, so now I'll talk about a case study where we generated both an immune library, but also an approach to generate a synthetic library, and then did phage panning against a really tough to drug target. And that target was ADORA2A. So I'll talk about that in a subsequent slide. But the approach that we used was to first of all do an immune library. In this case, we did a mouse immune library where we immunized either a humanized mouse or the Adveris diversimab mouse that I talked about before. We also did NGS analysis of those repertoires derived from those mice, and we actually used that NGS data to generate synthetic libraries that we could then pan and screen against the same target. And again, once we had those libraries, either immune libraries or synthetic libraries, we then did multiple rounds of panning against detergent-solubilized ADORA2A in this case to come up with those specific antibodies to that difficult-to-drug target. For the immunization that we did in the mice, we actually used a DNA-based approach to get to titer. So we were able to separate both the DNA immunization to get using native target, and then we used the detergent-solubilized ADORA2A for the panning work itself. And one of the leads that we generated was TB206-001. It actually came from the immune library campaign that we did in the humanized mouse. And what we found is this antibody has a very high affinity. It's specific binder to ADORA2A. And it's also cross-reactive with cynomolgus and mouse ADORA2A as well. And so shown here on the left is just the titration of the antibody against ADORA2A compared to a control antibody. We see very high affinity binding to this, again, really tough to drug target. This is actually one of the very first cases of a known discovery of an ADORA2A antibody. ADORA2A is very well validated in the small molecule space, but we're actually able to find an antibody to this really tough to drug target. Shown in the middle is just some cross-reactivity studies that we've done to show that the antibody has, that's very specific for ADORA2A, as well as shows cross-reactivity to mouse ADORA2A. And then again, shown on the right is also showing that we have nice binding to cynomolgus ADORA2A as well. We then did a whole series of assays in vitro to show that we could see potent activity. Shown on the left, we see nice inhibition of the ADORA2A pathway in a cyclic AMP assay. We've also recapitulated this in a PBMC assay. And then on the right, we also redid the same assay with different levels of activator in the assay. And we again see a nice correlation, titration activity with our antibody. Again, correlating that we have a high affinity antibody that blocks the pathway where we can actually see nice inhibition and activation against this immuno-oncology target. And then finally, we did an in vivo study. Again, this is in a human transgenic model using a COLO205 tumor. We see really nice activity of our antibody relative to for different isotypes. We also did a study where we compared its activity to an anti-PD-1 pembrolizumab control, where we actually see better activity of our antibody relative to pembrolizumab in this model. We actually don't see equivalent activity to a small molecule inhibitor of ADORA2A, and so we were continuing to see if we can improve the potency of this antibody through additional rounds of optimization. Okay, so now I'll talk about, again, a really exciting innovation in the team that we've also recently launched as a potential service that we're doing. This is actually the subject of another poster that we're giving, and which I'll give a list at the end of my talk today. But it's this idea of coming up with target-specific synthetic libraries that are actually based on a de novo design that we did in collaboration with a company called Macromoltec. And the idea here is that working with Macromoltech, they can actually design synthetic de novo antibodies that can hit multiple epitopes on targets. We gave them a really tough target, in this case C5a receptor, and they identified multiple epitopes on C5a receptor that potentially would be accessible to an antibody. We gave them a germline framework that we wanted a heavy chain, light chain antibody to be generated, the library to be generated from, and then they generated 10,000 putative binders to those multiple epitopes on C5a receptor. And then we then used those 10,000 antibodies to then generate a library where we focused and shuffled all of that diversity from those original 10,000 hits. So the idea is that you could potentially, if you wanted to, focus in on one epitope. For this particular case study, we really opened it up and then used multiple epitopes in that analysis. But the nice thing about this approach is that you could potentially, if you know what site you want to hit, we could potentially go in and design de novo antibodies to that one particular site, and then again, shuffle that diversity to really open up the space of potential sequences and binders to that particular target. So I'm really excited about this approach because I think it could really help us tackle really tough challenges in terms of specificity where you absolutely need to hit one particular site on a target through a library-based approach. And then once we have that library, we can then do panning. In this case, we used C5a overexpressed cells. Anytime we do cell-based panning, we actually use our NGS machine learning workflow that I talked about before to pick out specific hits against the target. Again, we can look at enrichment of specific clones as well as clustering analysis to come up with different families of sequences. And we can also generate deep learning models to pick out rare clones from our data as well. And we used all of those approaches to come up with the whole family of sequences that came from the panning work that we did. Shown here is just a little bit of FACS analysis where we then took those sequences derived from the machine learning lead picking. We made hundreds of different antibodies from that campaign, and we showed that we're able to see really high affinity binding to C5a overexpressed cells relative to the background cell line. This is just showing you FACS binding analysis at 100 nanomolar concentration. I'll show you later some data where we actually do a titration on those same cells. And that's shown here on the left. We had compared this actually to a clinical control C5a receptor antibody and showed really nice similar levels of affinity relative to the clinical control. We also did SPR binding using a recombinant C5a receptor that was made in mice cells by G-Clips. And we're actually also able to show for some of the clones high affinity binding against recombinantly expressed C5a receptor as well. We then looked at their activity in a beta-arrestin recruitment assay and actually showed that we had really nice functional activity for many of the clones. It wasn't at the same level as the clinical control antibody, and so that's why we actually went further and did additional rounds of optimization to see if we could increase the potency, which I'll show you in the next slide. One thing I didn't mention is that while we were doing this target-focused approach, we also did an approach where we made an immune library as well as used our own library of libraries against the same target. We were able to find functional hits from those other library approaches, but what we found is that this target approach for this particular target was very fruitful in allowing us to find even more hits relative to those other kind of more traditional approaches. So again, it's really exciting for us to think about other ways that we can use this in a very targeted, focused approach to come up with a target-focused library for a customer project. And a nice, another piece of data that we found is that all of the functional hits that we generated were not in the original de novo data set. We actually found antibodies where we had shuffled diversity from those original 10,000, but it wasn't like we just fished out that hit directly from the data set that we received from Macromoltech. So really the library-based approach coupled with those original 10,000 sequences was really important in helping us generate those functional hits. And then as I mentioned, we did take several of those leads forward to additional rounds of optimization. And through that library-based optimization approach that I talked about, we were actually able to find antibodies that now had very similar levels of functional activity relative to the clinical control. Okay, my last case study, I'm going to talk about how we can utilize a hybridoma approach to come up with an anti-ID antibody. This target is actually a bispecific antibody that has a Fab portion to it and an scFv portion to it. And we actually found anti-IDs to both portions of the bispecific. And we'll actually see that we can, the antibodies that we generate against either the Fab or the scFv both bind to the bispecific in a very specific way to each of those different portions of the bispecific antibody. So the overall workflow is, again, we took the bispecific, we actually broke it down into the Fab portion of it and the scFv portion of it, did 2 separate immunizations, and then we did hybridoma library generation and then ELISA-based screening from that approach. We then did a very thorough specificity analysis in ELISA, but then we followed that with BLI to get affinities. And then we, again, sequenced all of the clones that had the desired either Fab or scFv specificity against the bispecific. This just shows a little bit of work that we did on the titer. So again, we're trying to show specific titer for both the Fab-based immunization as well as the scFv-based immunization. Just shown on the right is just in the middle. Sorry, I keep hitting the wrong button. Where we get nice titer check that's very specific to either Fab or scFv, depending on what we're immunizing with. And then we did a very large primary screen, and then we did a subsequent secondary screen, again, looking in ELISA for that specific binding. And then from that secondary screen, we actually had over 182 clones per cohort, and then we did a nice either Fab or scFv-based ELISA screen to show that desired specificity and ranking. And then we pushed forward all the clones that had the desired specificity against either Fab or scFv. And then from here is just, again, a breakdown of the top binders. We looked at both binding to the bispecific targets. So again, if you had a specific binder to either the Fab or the scFv, you would expect both of them to have binding to the bispecific, which is what you see in ELISA. But here, again, you can see the Fab immunized cohort really only shows binding to the Fab portion, or the Fab alone. It doesn't show binding to the scFv. And then vice versa. All the antibodies from the scFv cohort show nice binding to the bispecific as well as the scFv. And this just shows you, again, some more secondary screening. We also did some screening in the presence of plasma, just to show that it could be used for samples derived from either an in vivo or human study. And again, this is really why customers are coming to us for anti-IDs, because they want to track their particular molecule in human clinical studies. And so showing the specificity in the presence of plasma was really important to them. And then again, this also shows you some of the affinity data from some of those leads. You can see that we're able to generate very high affinity binders to both the Fab as well as the scFv portion of the bispecific. And so this is, again, a really cool application of our anti-ID service. We were able to find binders that were specific to each portion of the bispecific antibody. So just in conclusion, we were able to find highly specific clones that were able to target the different portions of the bispecific in the presence of human serum. We found both blocking as well as non-blocking profiles, in the sense that we're able some anti-IDs actually block the binding of that specific target to that site, whereas there were other anti-IDs that didn't block the binding of the target to that site. And we were able to deliver back to the customer sequences, as well as the resulting vectors, as well as all the DNAs for those particular hits, and really showed the value of allowing them to move forward in their development project to track their molecule in clinical samples. So just to sum it up, I hope I've given you some nice stories around how we can utilize our technology in multiple projects. I mean, I showed you an example where we can discover VHHs from beacon-based approaches, immune library approaches, our library approaches. I talked about how we can use immune-based libraries either from mice or actually generate synthetic libraries from those NGS repertoires. I talked about how we can generate libraries in a de novo case, where we can actually generate a target-specific library, which, again, I'm really excited about launching as a potential service for our customers. And then finally, I went through a nice anti-ID case study where we're actually able to find specific binders to different portions of a bispecific antibody to really enable us to detect that antibody in human serum. So just in terms of, again, the power of Twist Biopharma Solutions, really that breadth of different technologies, we were able to see that in the multiple case studies. And and how we can tackle really tough targets by using multiple approaches. We really have high quality in terms of the people that we bring to the table on our projects, but also all the work that we do to generate the DNA as well as the resulting antibodies and all the downstream data. And then finally, we're really fast. We're really pushing the envelope in terms of how fast we can synthesize DNA, and that's enabled us to make and test our leads that much faster. But we're also doing a lot of work to optimize our workflow to actually automate the entire process of antibody discovery and antibody optimization. So that enables us to get back to you sequences and data that much faster as we do projects for our customers. And then finally, I did actually mention and describe a little bit on a high level some of the data that's actually in many of the posters that we're giving this week. We're actually giving 7 different posters, which are shown here. Definitely check out those posters if you want to get more information about many of the things I talked about today, but also some additional stories that I didn't tell today that are also, again, really exciting, great innovations within the Biopharma Solutions team, as well as within our libraries team as well. And with that, I'd be happy to answer any questions. Thank you for your time and attention. Yeah, the question was, has Twist ever thought of doing non-customer internal projects to generate our own assets for licensing? Yes, we have. We've done a lot of proof of concept work with our libraries as well as our in vivo approaches, really to validate our tech. And as a result of that, we actually have several tiers of validated assets available for licensing. So if anybody's interested in what we've been able to come up with over the years, please follow up with us, and we can send you a list under NDA. Yeah.
Very interesting talk. I had a question on how you would approach with the AI modeling for these rational designs that have multi-epitopes within a molecule. How do you approach this when there's not a clear crystal structure? What are you going to base this on? There's a lot of unknowns in biology, and half the stuff that's out there in some of these crystal structures are either incomplete or roughly based off of some variant that has been partially crystallized and then
right. Right. So the question was, for the de novo design, did it require a crystal structure to kind of design those de novo antibodies, which may or may not be a good representation of the actual structure? What other approaches could we potentially use? I think we also can use modeling-based approaches to come up with, to kind of complement the crystal structure. I think the modeling-based approaches for target structures is getting more and more powerful, as you probably know. And so we can either use an input model or we could use an actual cryo-EM or crystal structure as the input, and we can actually compare them all too to make sure that they're in agreement with one another, to make sure that we have the right structure to start with for our de novo antibody discovery work. Yeah.
So the prediction that you put there, there were two, for the developability you put 2 criteria. One was the heat retention, which I guess you can validate in the lab easily.
Yeah.
Or relatively easily. The second one you put the immunogenicity prediction. How do you validate that?
Yeah, so we can use we can try to use experimentally determine it. I think we're thinking about maybe doing some in vitro-based, cell-based assays as the input data set to then correlate back with in vivo clinical immunogenicity. Of course, it's really hard to input human data into that process. We'll try to, but I think we'll probably start with cell-based functional assays as our input, where there's known precedence in the literature that we see a nice correlation with in vitro versus clinical outcome.
The immunogenicity prediction is how human-like the molecule is?
We always look at the level of humanness in the molecule. We can always determine that, and we can also see how that correlates with immunogenicity. And I think what we see in the literature is that after a certain point, it does correlate, but initially there's a lot of scatter in the data in terms of that correlation with a level of humanness. Yeah. Yeah, you.
For the C5a, is the intention to generate binders or agonists, or agonists byproduct of binders?
The intention was just to develop binders, because in this case, we didn't really know what the functional epitope was, because ideally you want to hit right inside the middle of the cavity of the GPCR, but As you may have seen, we really looked at epitopes around kind of the outside rim of the GPCR, because that's really what's accessible for the antibody to bind to. So we really didn't know a priori what the functional epitope would be. But again, as I mentioned, if you do know, if you really do have a good understanding of where on the target you want to bind to, we could really home in and look specifically at that site. Yeah.
Aaron, for the TKD1 project, you've shown that in your cohort, vast majority of your clones, VHH clones, were binding to the different epitope that the clinical molecule was.
Right.
Now you found one. Do you know why they chose that one? Because typically, if you look at the immune library, the most predominant epitope is what vast majority of people are getting.
Right.
Why did they select that one? Do you know?
Again, I don't know exactly, but it probably came from another, I would assume probably an immune-based approach. They probably went through either mice or maybe a humanized mouse. And so that probably was the dominant epitope for them that came through their approach, whereas when we did phage display using a synthetic library, our dominant epitope was the other site.
This one was from synthetic.
Yes, yes. Great, so again, thank you so much. My slides will be publicly available, so if you want to download them, please feel free to do that. And then also, again, check out all the posters that I talked about. There's a lot more data, particularly for some of the stories that I gave, so definitely check out our posters tonight and tomorrow. Thank you so much for your time and attention.